So today we are going to talk about chemical analysis of bone and the evolving consumption of maize in the Ecuadorian Andes. And I was planning to introduce myself, but Kevin already did that. Still, I will just mention that I'm an archaeologist who have worked on several excavations in Ecuador and have also experience with the analysis of artifacts such as ceramics, obsidian, marine shell. Most of the time I work as a biological anthropologist doing the analysis of human remains. So I work on estimations of sex, age, ancestry, stature, pathologies, trauma. And I've worked in Ecuador and in China. And I also work as a forensic anthropologist. And I have participated in police searches in Canada and in forensic workshops in Canada and in Ecuador. So now moving on to what we are going to discuss today, um, we are going to talk about diet. So we have this first question: what elements are often considered archaeological evidence of human diets? And we start first with animal remains. So animal remains are part of what we call the zooarchaeological record, and they are examined by zooarchaeologists who are trained into identifying the species of these animals, right? Sometimes we are lucky and we find the whole skeleton. Sometimes we only find a few bones. And sometimes it gets even more challenging and we have to identify a species in an archaeological site only using tools that were made of bone, right? Then we have the plant remains. So these include examining carbonized seeds, pollen, and phytoliths. Phytoliths are microscopic structures made of silica that are found in some plant tissues that persist long after the plant has decayed, right? So today I can go to an excavation, I can take some samples of soil, and then I do a flotation technique. And after that flotation technique with water, I can still find some of these um, plant remains that I can take to the lab and examine with a microscope, and I can identify which plants were available at that time. So here we have an example of pollen seen under the microscope, and also of maize cobs that have been found in archaeological sites, right? And it's very cool the level of detail where we can even see the size of the maize cob and probably establish how important were these maize cobs or these maize in the diet. Then we have lithic and ceramic artifacts. So let's start with the fact that the function of the artifacts can also give us information about diet. For example, if the tools were made for cutting meat, for prepping skins, or for fishing, or if the vessels were made specifically for containing liquids or solid foods. In addition, the surfaces of these tools or these vessels can still contain food residues. So just to give you an example, the last photo here is from two vessels that were found in the Amazon region of Ecuador. What is cool about this vessel is that inside it we found the most ancient residues of cacao that have been found yet in the world. And they are dated to 8,000 years ago, which is very cool, right? Um, Other elements that give us information about diets are iconography and representations in artifacts. Examples include ceramic representations and depictions of food-related images or symbols. So sometimes archaeologists can encounter ceramic vessels that have the exact same shape and appearance of fish, of mammals, of birds, of squash, of other fruits, right? And that give us information about the type of animals or fruits that people know back then, right? Here in this picture, for example, we also have an image that was found in a ceramic vessel in Peru, and it is from the moche culture. And you can see here that these people are sharing a cup, probably filled with a drink made of fermented maize, which we call chicha. And we also have the depiction of a dog, right? So we know that these people have dogs at that time. Next, we have texts and chronicles. So texts such as chronicles written by travelers give important information about past ways of farming, cooking, and eating, among other practices. So for example, here in Ecuador, we were colonized by the Spanish. And Spanish travelers came and wrote a lot about our funerary practices and other practices related to food. So that's a good source of information as well. We also have human bones and teeth. So signs of malnutrition and stressors can sometimes be seen in bones and teeth. So we can see the lack of certain foods in the bones, right? Or the lack of actually food in general. So we can see it in the way of anemia. So in the first picture here, you can see some porosity in the rooftop of our orbitals, right? And that shows anemia. So we can see that this person was struggling to get all the food nutrients he or she needed. Another example is that the excess of starches and sugar in the diet can lead to dental caries, like the photo that we have here. Um, so that gives us information about diet as well. For example, in the Preceramic period, long before agriculture and long before eating maize, people used to have very low frequencies of caries. But after agriculture and very high consumption of maize has made these the frequencies of caries become much higher. Finally, we have chemical analysis. Chemical analysis such as the carbon and nitrogen stable isotope analysis provide more insights into diet. And we are actually going to focus today on this type of analysis. But some of you may have heard or not about stable isotopes. So let's start with what are isotopes? Isotopes are atoms of an element from the periodic table that have an equal number of protons but a different number of neutrons. This means that they have slightly different weights, and they can be of two types. They can be radioactive or they can be stable. Radioactive isotopes decay over time. An example is carbon-14 or C14, which is used for radiocarbon dating. Stable isotopes, on the contrary, do not decay, and their abundance does not change over time, which is excellent for us who when we are trying to measure them. Examples are carbon-13 and nitrogen-15, which are used for studying diet. Isotopes are present everywhere, but the ratios vary between different substances. For example, different types of food will display different isotopic ratios of the same element. And the same happens with ecosystems. So, for example, elements present in land, in sea, or freshwater environments will display different isotopic ratios. Or elements in different climate zones will also show different isotopic ratios between them. The isotopes that are in the food we eat and the water we drink are being constantly incorporated into all our body tissues, including our skeleton. So for example, if I want to study the hair or the nails of a person, I will be able to get information of the last weeks or months or even a year of that person's diet. If I choose to study the ribs of a person, I will get information about the last decade of the diet of that person. And if I choose to study long bones of a person, I will I will get information of the last 30 years of the diet of that person. Now, by measuring the ratios of carbon and nitrogen stable isotopes in human and animal bones or teeth, we can trace those ratios back to the original food resources they consume. And what these original food resources are often plants, right? Because humans, like animals, eat plants, and human eat plants, and human eat also animals that have consumed those plants, right? So everything goes back to plants. Now, so before we start talking about the isotopic signals of animals and humans, we start with basic notions about the isotopic signals of plants. So we can basically study two groups of plants: the C3 plants, which follow the Calvin-Benson cycle or the C3 photosynthetic pathway, or we can study the C4 plants, which follow the Hatch-Slack cycle or the C4 photosynthetic pathway. Now, I'm not going to get into much detail here, but we all know basics of photosynthesis, right? So going back to C3 plants, they exhibit, let's remember here we are going to talk about carbon 13, right? So they exhibit carbon delta 13 values that range from minus 33 to minus 23 per mil. That's their isotopic signal in general. And they represent about 95% of all plants in the planet. Now moving to the C4, examples of these C3 plants are cereals, potatoes, quinoa, like pretty much every single plant that you can think of. Now moving to the C4 plants, they display carbon delta 13 values that range from minus 16 per mil to minus 9 per mil. And what is cool about C4 plants is that there are not that many. We have maize, we have sugarcane, we have millet, we have sorghum, and some types of amaranths, but that's it. Finally, we have the chem plants, and they're not very important for the interpretation of diet because mainly they are represented by succulents. But what is cool about chem plants is that they can use both C3 and C4 photosynthetic pathways depending on the environmental conditions. So for example, if they're in an area that is more deserted, that doesn't have much water, they will try to use C4 photosynthetic pathway. Okay, so here in this graph, you can actually see how we see the isotopic signals of plants, animals, and humans. So if we focused in this lower part of the graph, we can see that here we have the carbon 13, the delta carbon 13. And on the other part of the graph, we can see the nitrogen delta 15. If we focus right now only on the carbon, we can see that here we have our C3 plant, right? About maybe minus 22 of carbon, right? Next to it, we have an herbivore that is only eating C3 plants. So their isotopic signal is very similar. The same happens with other herbivores. Now, we can have some carnivores that are eating these herbivores that have only consumed C3 plants. And that's why all their isotopic signal is kind of similar. Here, on the other hand, we have as our C4 plant. In this case, is maize. We can see that the isotopic signal is about minus 6. In this case, any animals or humans that are only consuming maize will have a very will have an isotopic signal that is very close to this one, right? Now, if we have a human or an animal that is eating both C3 and C4 plants and is kind of in a balance, the isotopic signal will be here in the middle. Now, let's move to the nitrogen. The nitrogen depends a lot on the trophic level. So you can see here that the carnivores are a trophic level above the herbivores. So that's why they have a higher nitrogen 15 level. The same happens with humans, because humans eat protein from carnivores or herbivores. We have a higher nitrogen delta 15 value. So basically that's what we try to see here. Now let's move to our study case. So it will be about a studying diet in the formative period using carbon and nitrogen stable isotope analysis. And this was part of my master's thesis research. Here I focus on analyzing human samples from Rancho Bajo, Kotoku, and as orchideas. Now, before we start, I know that none of you are familiar with time periods in Ecuador, so I will just explain some basics. Let's start at the bottom here with the Preceramic period, which is the oldest period. It started about 8,800 years before Christ. And of course, as the name states, it is characterized by the absence of ceramics. Then we move to the formative period, which is more important for us today. The formative period, you can see that it has two different dates, 4,400 and 2,400. The first date belongs to formative sites in the coastal region because this period started earlier there. And 2,400 is when the formative period starts in the northern highlands. So this period is characterized technically by the appearance of ceramics, by agriculture, and by villages. However, in recent years, we have found evidence that there were archaeological sites that belong to the formative period, but do not have any evidence of ceramics, right? So we are still trying to work on our definitions of every time period. Then we move to the regional development period. This is a period that is kind of tricky because we know about it in the coastal region, but again, we don't have much information in the highlands. What we know is that in the regional development period, there was more social stratification, so that we have people that were elite, and we have people that were non-elite, and their diet was very, very different. Then we move to the interration period. The interration period was characterized by a lot of population density. We have also a lot of commerce, people that were traveling throughout all the territory. There was a lot of connection between the Amazon region and the coastal region and the highlands. And we have also human settlements that were fighting what came later, which was the Inca conquest. The Incas came from Peru, and they stayed in the country only for about 100 years. Later, after the Inca, we were colonized by the Spanish. So let's start with what is the earliest evidence of maize in the Ecuadorian Andes. In the Ecuadorian highlands, maize pollen and phytoliths from sediment cores of Lake San Pablo, which you can see in the picture, have indicated the presence of maize as early as 4,000 years before Christ, long before the early formative period. So because we have this early evidence of maize from the Preceramic period, archaeologists started to think that it was obvious that people will eat it in the formative period, and from then, its consumption will just start increasing over time, right? It will increase throughout time. But we will see if that's actually true or not. I will start with just giving a few details about the sites that I included in my study. Starting with Rancho Bajo. Rancho Bajo is an archaeological site that was discovered in 2011 in the city of Quito, where I am from, in the northern highlands of Ecuador. To this day, only 28 burials have been discovered. The majority of them are individually primary burials. This means that each tomb have only one individual, and none of them were starved until the archaeologists discovered them, right? So nobody touched them until we found them. Then we also have that no faunal remains were found at the site. We don't have habitational contexts, and we don't have ceramics. The only thing that we found at this site were lithics, which were made of obsidian and basalt. Because of all this evidence, we started to think that this site was a hunter-gatherer site, and that it was confusing, right? Because the radiocarbon dates place it in the formative period, but all the other evidence suggested it was a preceramic. This site was from the Preceramic period. But recently, we conducted a stable isotope analysis on some carbonized seeds, and we found evidence that there was maize at this site. So probably they were cultivating maize, which is very interesting. Then we have Kotoku. Kotoku is one of the most famous formative sites in Ecuador. It was discovered in the 1970s. And what we have found here is that there were two settlements, an early settlement that belongs to the early formative period, and we have a late settlement that belongs to the late formative period. At this site, we have found a lot of ceramics, a lot of lithics, lots of faunal remains from rabbits and deer, especially in the early settlement. In the late settlement, we also found remains from camelids and guinea pigs, and lots of birds. We also found carbonized seeds of maize and beans, and evidence of chocha, which I will show you a picture later if you're not familiar with it, and quinoa, potatoes, and beans. 13 burials were found in connection with the early settlement, and about 200 burials in connection to the late settlement, which shows a lot about how the population density increased over time in this same site. Finally, we have Las Orquídeas. Unlike Rancho Bajo and Cotocollao, Las Orquídeas is a bit far away. It's about 100 kilometers away from these other two sites, and it was discovered in 2013. Among the evidence that we found at the site are a hearth, a trash pit, and 8 human burials, which means that people were not only buried here, but they also lived there. We also found a large body of craft production, and we have artifacts made of a great variety of raw materials, like mother of pearl, like marine shells, spondylus, which is considered an exotic good. It's basically a red marine shell, but it had a greater importance in the Andes. We also found these two ceramic vessels that you can see here, the drawing. And what is interesting about these two ceramic vessels is that they were the only grave goods that were found at the site, which means that only one person at the site, which was a young female between 14 and 17 years old, was found buried with those two vessels, which suggests that this probably was an important person for the settlement, right? And we also have a lot of animal remains. Some of them were guinea pigs, birds, camelids, deer, deer, rabbits. So here we can finally see what are the isotopic signals of the people from Rancho Bajo. The people from Rancho Bajo are represented by these red figures here. And what we can see is that their isotopic signal is about -19, -18. This suggests that they had a diet that relied more on C3 plants, but it still had some maize consumption, right? Very modest amounts of maize in the diet. Something else that is interesting is that you can see that the signal of all these people is kind of clustered together. This suggests that these people were sharing everything they eat. So if someone killed an animal, they were eating all the same protein. If someone was like gathering all these plants and fruits, they all eat the same. And that's why they all have the same isotopic signal. Here we have the isotopic signal of the people from Kotoku. So here in yellow, you can see just one triangle, and that represents the only human from the early settlement that I was able to sample. However, it is interesting that it has the same isotopic signal from the people from the late settlement of Kotoku, right? And they all have a similar signal with the Rancho Bajo. This suggests that they still rely more on C3 plants and only small amounts of maize. However, because their signal is not that clustered together, we can suggest that their diet was more varied. And it's not a surprise because remember that in the late settlement there were about 200 burials. So it suggests that a lot more people were focusing on gathering food resources for the settlement, right? In terms of the animal protein and their nitrogen values, we can see that their isotopic signal is above the signal of the rabbits and the deer, which were available at that time. And in terms of the late settlement, their eye signal is also above the camelids and the deer, right? So they were probably feeding from all those animals' meat. Here we have the signal of Las Orchideas. And Las Orchideas has a very different isotopic signal, which suggests a lot more maize consumption in comparison to the other two sites. In addition, we have the isotopic signal of this single individual, which, if you guess, is that young female between 14 and 17 years that was buried with 2 ceramic vessels. This probably suggests that this young woman came from a different settlement and died at Las Orchideas. And also that probably she was very important at the settlement. This also means that there were settlements at the same time from Las Orchideas and late Kotoku that were eating a lot more maize. Another interesting finding is that these 2 deer that are represented here in yellow have an isotopic signal that is more similar to the people from Las Orchideas. So probably these 2 deer were eating maize, or probably the people from Las Orchideas were feeding them, like domesticating them with maize. So that was it for my thesis research, but I wanted to compare my data to 2 other studies. So the first study is about the site La Florida. La Florida is also in the northern highlands of Ecuador, and to some archaeologists it belonged to the regional development period, but to others it belonged to the integration period. What matters here is that we have the isotopic signals of high status people and low status people, and they all show more maize consumption than the people in the formative period. So until now, we can see actually that there is an increase in the consumption of maize from the early formative to the late formative and to the regional development or integration periods. Here is the data from the second study I used to compare with my data. Let's focus on the blue dots. These blue dots represent a site called Tajamar. What is interesting is, again, that we see that more maize consumption, even more maize consumption in comparison to La Florida. But if we focus on the second site analyzed by this study, which is NAIC, which stands for the New International Quito Airport, where these remains were found, we can see that the isotopic signal of that people is extremely similar to the people from Rancho Bajo that were from the early formative period. So this clearly shows us that we were very wrong to think that every sediment starting from the early formative started to consume more maize throughout all the subsequent time periods, right? We can see that there were different subsistence systems at the time, and not everyone was eating that much maize. So maize wasn't as important as we thought for every single settlement, right? So basically the results are that from the formative to the integration period, there was an increase in the consumption of maize. However, this increase was not seen in every settlement. For some of them, maize was not an important food staple at all, and there were different subsistence systems, as I mentioned before. These findings contradict to a certain degree previous interpretations about diet, where maize played a major role in diet, and these interpretations were only based on the animal remains and the plant remains that were identified at these sites. So they found lots of maize cobs, lots of pollen, and they thought, yes, these people are eating a lot of maize, but actually, no, they weren't. And here are some aspects that I want you to consider in the future. First is that identifying the presence of a specific plant in a site or region does not necessarily mean it was consumed or it played an important role in a particular society. Second, that to have a better understanding of diet, we really need to combine the analysis of zooarchaeological and palaeobotanical records with the stable isotope analysis. And that analyzing data from different sites and different periods at the same time altogether gives us the opportunity to identify dietary trends and changes that took place over millennia. So that's it. Thank you much. I don't know if you have any questions.